So, in our current video, we are going to be talking about the first half of meiosis, and we might get to meiosis too as well. So, because we needed to get 4 cells by the end of meiosis, we're going to go through 2 rounds of cell division. We're going to go through meiosis 1 and then meiosis 2. So, the first phase of meiosis is prophase 1. This will look really similar to mitosis, but the biggest difference is the thing or the process that happens at the end of prophase 1, which is called crossing over. We have all the same trappings. The nucleus breaks down, the chromosomes condense, the spindle fibers form. All of that stuff still occurs. But we have this process called crossing over where the homologous chromosomes begin to pair up, and once they pair up, crossing over can occur. What crossing over is where the homologous chromosomes actually exchange sections of their DNA. So we have our 2 homologous chromosomes here. The legs or the arms of these chromosomes are going to overlap. They're going to actually exchange sequences of DNA. This is one of the first ways that we can get genetic diversity into our gametes. This is called genetic recombination, and it increases genetic variation and genetic diversity in a species, because now if one of our offspring ends up being created from a gamete where we had a crossing over event, they get an entirely unique sequence of DNA that has not occurred before. So then we have metaphase. Metaphase 1 is where the chromosomes are going to line up in the middle of the cell, which sounds pretty familiar, except when you think about how they're lining up. In metaphase 1, in meiosis 1, we're going to line up as homologous pairs. That means we're going to have each of the pairs of chromosomes standing or lining up next to each other. This is very different from mitosis, where they lined up in a single file line so that we could pull apart the single chromatids from each pair of chromosomes. Now, one of the big things that happens here is another way we get genetic diversity, which is called the law of independent assortment. That states that these chromosomes are going to line up on either side of this middle point here, this middle line, regardless of where they came from. So you could end up with all of your mom's chromosomes here on the bottom, but you could have some mix of your mom and dad's chromosomes on either side of this dividing line. That is called the law of independent assortment. So we could have a mix. So this could be our mom's chromosome, and this could be our dad's chromosome. And when you start to think about organisms like ourselves, where we have 23 of these homologous pairs, all of which are lining up individually without any regard to whether they came from our mom or from our dad, you can start to see how we can really increase genetic diversity. So we've covered 2 ways to get genetic diversity so far. We've talked about crossing over, where we switch sections of a chromosome for another, and the law of independent assortment, where our chromosomes will line up regardless of where they came from. The third way we can get genetic diversity is through a process called mutation. When we go through DNA replication, one of the things that can happen is our chromosomes may not be replicated properly. So we may get errors in the DNA sequence. Those errors are called mutations. And sometimes those mutations will be passed on to our offspring, and those mutations may be beneficial or detrimental, but they are different than the original sequence. So one thing to make sure you have in your notes is the 3 ways we get genetic diversity: crossing over, the law of independent assortment, and mutation. Then we have anaphase. Anaphase is where we pull apart the chromosomes. This should look significantly different to anaphase in mitosis because we're not separating chromatids here. We are pulling full chromosomes to opposite sides of the cell. Then we have telophase 1. Here we have our chromosomes fully separated. They're now in different cells, and these cells have separated so that we can begin our next round of division. Now, I just want to compare where we started and where we ended up. So here we have metaphase, where we still have all 4 chromosomes in a single cell. Now we have the end of telophase 1, where we have 2 different cells, and we've already set up that haploid result because we only have one version of each of the chromosomes. We only have the green version of the big chromosome and only the red version of the little chromosome, and the opposite is true down here. These will not be replicated again. We will not go through another round of DNA replication. We're going to divide these cells as is. So here's meiosis 2. We're going to divide both of these cells in order to get our 4 gametes at the end. So the nuclear membrane breaks down. This is prophase 2. But again, one of the big differences here is that we have 2 cells doing this division. Metaphase, the chromosomes line up in a single file line, but again, it's spanning 2 cells. So it looks similar to mitosis, but we have 2 cells doing it, which makes it drastically different than mitosis. Anaphase, the chromatids get pulled apart to opposite ends of the cell. And then telophase 2, where we have the final result of meiosis, where we have 4 independent, genetically unique haploid cells. And it doesn't matter how many times we do meiosis, we're never going to get the same cells. They are always going to be 4 genetically unique cells, no matter what way we do meiosis, because we have crossing over occurring and the law of independent assortment and mutations occurring. So every time we do meiosis, we're going to get unique sets of cells, even from the previous round of meiosis. I'm going to leave off our notes here. We have one more topic to discuss that's related to meiosis, but I don't think we have enough time to really get into that for this video. So this is where this video is going to end.